Hi guys. So as medical students, we hear a lot about immunoglobulins and all their different functions. So it's probably a good idea to get to know them a little bit better. So before we get into the different classes of immunoglobulins, let's quickly go over what they actually are. Immunoglobulins are essentially antibodies, which are globulin proteins. You can remember them as having a globular structure or a quaternary structure, and they're released by plasma cells or B-lymphocytes. But in order to understand immunoglobulins, we must go back to its origin, which are the B cells. Well, B cells or B lymphocytes are developed in the bone marrow in response to signals from stromal cells. The first recognizable stage of B cell development is the progenitor B cell, or pro-B cell, which goes on to express stem cell associated antigens like CD34 and CD117, and as well as B cell line specific antigens such as CD19 and CD22. Immunoglobulins, however, are synthesized in the latest stages of development, and IgM is the first to be detected by its heavy chain in the cytoplasm of the pre-B cells. Complete IgM immunoglobulins can be found at the surface of the B cells that have not yet come into contact with any foreign antigens. Now, these cells are typically known as virgin B cells. Now, the other immunoglobulins are differentiated only after coming into contact with specific antigens, and they are found as receptors on the surfaces of mature B cells. So, as a quick rundown, there are 5 classes of antibodies. We have IgG, we have IgM that we were talking about earlier, we have IgA, IgD, and IgE. Now, the antibodies are subdivided into these 5 classes based on differences in their heavy chains. Now, you're probably wondering, what is all this talk about antibodies and chains? So let's take a look at the structures of these immunoglobulins just to get things straight. The simplest antibody molecule has a Y shape and consists of 4 polypeptide chains, 2 light chains or L chains, and 2 heavy chains or H chains. The terms light and heavy refer to the molecular weight in the range of 25,000 Daltons and 50,000 to 70,000 Daltons respectively. There are 2 types of light chains, denoted by kappa and lambda. The heavy chain contributes to Fc and Fab regions, while the light chain contributes only to the Fab region. The H chain symbols for each class of immunoglobulin is different. The symbols are shown as gamma for IgG, alpha for IgA, mu for IgM, theta for IgD, and last but not least, epsilon for IgE. Now, the Fab, or the variable regions of both the light and heavy chain, are responsible for antigen binding. You can remember this by the term itself, which is Fab, for fragment antigen binding. Whereas the Fab and Fc region of the heavy chain is responsible for various biological functions, such as complement activation, which you can remember by the C in the Fc, and binding to cell surface receptors. Keep in mind, however, the C in Fc actually stands for crystallizable. The Fc fragments possess binding sites for the complement factor C1q. But where do the names Fab and Fc really come from? This was actually done by an experiment conducted using an enzyme called papain. The papain separates 2 identical antigen binding, or Fab fragments, from 2 non-antigen binding fragments, or Fc fragments. This creates 3 fragments, of which the 2 fragments will still bind to the antigen, which is Fab, and the rest would crystallize, hence the name Fc. Another proteolytic enzyme, which is pepsin, which you may already be familiar with, acts by digesting the Fc portion at multiple sites. The 4 chains are actually linked by disulfide bonds that are formed between cysteine residues in the individual chains. Although binding between antigen and antibody involves only non-covalent forces, and therefore it is theoretically reversible. But in practice, the high affinity of most antibodies mean that they rarely become detached from the targets before they are destroyed. So coming back to the structure of the antibody, an individual antibody molecule always consists of identical H chains and identical L chains. The L and H chains are then subdivided into variable and constant regions. Now if we look here, the light chain consists of one variable or VL and one constant or CL domain. Now most heavy chain consist of one variable, which is VH, and 3 constant or CH domains. The IgG, IgD, and IgA have 3 CH domains, whereas IgM and IgE have 4. Now the variable regions of both L and H chains have an extremely variable or hypervariable amino acid sequences at the amino terminal end that form the antigen binding site. Both variable and constant regions consist of about 110 amino acids. Remember that the different domains of the given immunoglobulin molecule have a similar globular structure. This is characterized by the presence of beta-pleated sheets and disulfide bonds. Now, the remarkable specificity of antibodies is due to these hypervariable regions. Now, I keep talking about hypervariability, but what does this exactly mean? And how does it work with antigen specificity? Let's find that out. Now, this hypervariable region consists of 6 to 8 amino acid around position 30, 50, and 93 of the light chains, and around positions 32, 55, and 98 of the heavy chains. Now, these are the regions that determine the specificity of antigen binding, and they're usually referred to as complementarity determining regions, or CDR. Now, the substitution of a single amino acid in this region is crucial for the binding of a particular antigen. The interaction with specific receptors, however, and the degree of complement binding is determined by the constant region. Again, if we recall, immunoglobulins are proteins, and their amino acid sequence can be immunogenic for different individuals and different species. So they can act as an antigen. In fact, they can even act as self-antigen. Now they have isotypic, allotypic, and idiotypic variants. The isotypic determinants are responsible for the amino acid differences in the Fc regions and subsequently between immunoglobulin classes and subclasses, and between heavy chains and light chains. If we talk about allotypic determinants, they are the variations in the constant regions of immunoglobulin of the isotype, owing to the allelic variation in the genes found among different individuals. Now, idiotypic determinants are the individual determinants of any given antibody molecule in accordance with the variability of the immunoglobulin's CDR region. The blood mainly contains 3 types of globulins, which are the alpha, beta, and gamma, based on the electrophoretic migration rate, which refers to the migration and separation of charged particles or ions under the influence of an electric field. The IgG immunoglobulins migrate into the gamma globulin fraction, while the other immunoglobulins, especially IgM and IgD, have a lower electrophoretic mobility and are found mainly in the beta globulin fraction, but also in the alpha 2 fraction. So moving forward, what are we going to discuss in this lecture? We're going to go over the immunoglobulin gene organization, and we're going to discuss the individual 5 classes of immunoglobulins and their important functions. So stick around with us.